Hello, everyone, and welcome to today's webinar entitled Neuropathology of SARS-CoV-2 as Revealed by Spatial Biology. This webinar is part of the LabRoots virtual event series on the coronavirus. I am Elizabeth Schneider, Senior Global Marketing Manager at NanoString, and I'll be your moderator for today's event. Today's educational webinar is presented by LabRoots and sponsored by NanoString. For more information about NanoString, you can go to our website. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Susanne Krasemann. She is a group leader at the Institute of Neuropathology at the University Medical Center, Hamburg-Eppendorf, UKE. She's also the head of the core facility of experimental pathology, or the mouse patho lab. A little bit more about Susanne. She was a neuron-centric focused researcher for neurodegenerative diseases, but it became increasingly obvious in her work that other brain types actively influence disease processes. So the focus of her research is a study of inflammatory profiles in neurodegenerative disorders, including Alzheimer's and prion diseases. As the head of the core facility of experimental pathology, she performs high-resolution staining and imaging of pathological tissue samples. And as part of this work, she participated in several projects to study viral diseases such as Lassa or Ebola in mouse models. And this obviously led her into the study of pathogenesis of SARS-CoV-2 and COVID-19, especially in the brain 2 years ago. And she has already published a couple of manuscripts on SARS-CoV-2 abundance and their consequences in several tissues. And also her work that she will be presenting today has also come out in bioRxiv and has the distinction of being the 100th publication on the GeoMx digital spatial profiling platform. So thank you so much, Susanne, for being here, and thanks everyone for being on the line to listen in.
Yeah, thank you very much, Elizabeth, for this very nice introduction. And you already introduced me so nicely. So I will directly start with my talk. And today I would like to talk about the neuropathology of COVID-19, because, I mean, the thing is, COVID, as a name, is a disease of the lungs, and it comes with fever, fatigue, and cough, and also with like other signs of common cold. But frequently, we also see neurologic dysfunctions like the very common loss of smell and taste, but also more severe things like seizures, stroke, and others. And also patients that had COVID very often suffer neurologic problems, such as long COVID with brain fog, headache, but also, again, with more severe problems. So already very early into the pandemic, we asked what's going on in the brain. And since you all know that I'm at the Institute of Neuropathology in Hamburg, we were obviously interested about the neuropathology of the disease. And Hamburg is a city, but it is also an own state. And to get more information on the pandemic, the state asked the people from legal medicine to perform autopsies. So this was done really via a quick law. And thus, here in Hamburg, we had more than 300 full autopsies of COVID patients since March 2020. And we in the institute already investigated more than 40 brains of these patients already very early in the disease. And these data were already published, so we checked for common markers of that you also have upregulated or dysregulated in neurodegenerative diseases such as GFAP, which is a marker for astrocytes, or HLA-ADR, which is a marker of microglia, the innate immune system cells of the brain, or like CD8, which is a marker for activated T cells. And what you see here is that astrocytes do not play such a big role, obviously, in COVID-19. However, for the microglia, we could see, and this is marked by the green arrows here in the second column, that we saw some nodules, and also CD8, like T cell wise, we sometimes also saw T cells. However, when you check like other brains of people that died of independent causes, then you also see these kinds of like immune cells. So the question was how specific was this for COVID-19? And here you see an overview of the patients that went into this study in 2020. And the upper row is the astrocyte activation, the middle row the microgliosis, and the lower row the cytotoxic T cell infiltration or abundance. And more to the right side are the cases that were positive for viral RNA, and to the left side you see cases that were negative for virus via RT-qPCR. And when you check this table, you see that there's a lot of diversity within the samples, and there's not a real trend towards there is abundance of, or there may be the abundance of virus in the brain and more severe pathology. So we were really wondering whether the changes that are seen are more subtle, and thus we also went into close-ups. And what we could see here, and I already mentioned this before, that we saw this microglial nodules, so areas where you see clusters of these specific immune cells, and applying another marker, which is CD68, a marker for activated microglia. We saw that this is also usually also accompanied by activation. And what we could also see is that these clusters also are around vessels. So this was a question whether, and this was also one theory, because as the virus is like distributed via the bloodstream, so obviously the linings of the vessels would be the first contact site for the brain. And so we were interested to see, okay, is there anything going on with the vessels in the brains of COVID patients? And moreover, we also were very interested to see whether there's really virus detectable in the brain. And for this, we were interested to establish a really reliable staining for SARS-CoV-2 infected cells. And we started here with the vero cells that were really artificially or in the lab infected with SARS-CoV-2. And these cells are here marked with red arrows. So it's always a red column of the image are the infected cells, and the left column are then the uninfected cells. And we tested 13 different antibodies against SARS-CoV-2, against different proteins here. They were either directed against spike protein, against non-structural protein 3, against the double-stranded RNA, or against nucleocapsid protein of SARS-CoV-2. And you can very easily see that there are antibodies around that do not show specific bindings to infected cells. Then there were antibodies around that also show high unspecific staining in the uninfected cells. But then there were also cells or antibodies that worked nicely, so what you would expect, so uninfected cells. And these here are formalin fixed, paraffin embedded, so the same way as we would fix also tissue samples. And so we thought, okay, these antibodies that are marked with a green circle here would be suitable because they do not show any staining in uninfected cells, but show really high reactivity in the infected samples. And with this, we went to lung tissue and only focused on the antibodies that already worked in our cell-based system. And again, here, we could see that some antibodies performed better than others. And here, the infected or the lung tissue from a person with COVID-19 is marked in red, and lung tissue from like a non-COVID patient is non-labeled. So you see that, again, some antibodies work nicely, some produce high background, and some do not produce specific staining. But what was also obvious is that obviously antibodies directed against spike protein showed less signal than those of nucleocapsid protein. And and one other point is, and here's an example of a respiratory mucosa, that the presentation of infected cells is rather with a planar staining and not with a punctuate staining, as might be in some publications. But to further evaluate the specificity of the spike antibodies, we also used other spike antibodies that are directed against the receptor binding domain, against S1, and the antibodies that I already showed was against S2, in contrast to the monoclonal. And here again in the cell system, they seem to work equally good. But here again in the lung, and these are consecutive sections of a lung tissue, we again could see that the abundance of spike protein is much less than nucleocapsid protein. And this led us also to assume that detection of nucleocapsid is much more well suited for the detection of infected cells that is spike protein here. And because you detect rather more cells with nucleocapsid directed antibodies. And this could be further evaluated in infected virus cells in the upper row, again respiratory mucosa and lung from a patient that died of COVID-19. And again here in these cells, we could see that spike is much less. So our staining were usually then performed with nucleocapsid directed antibodies. And here's an example of lung tissue, and you really see how heterogeneous the picture is in a tissue where everybody thinks, yeah, this is the main target of the disease, so there should be tons of virus. And believe me, this is not always the case, because also these are examples of patients that died of SARS-CoV-2. So there might be only single cells infected, even in lung tissue. And furthermore, like tissue from people that might also have immune activation and other sequelae is prone to also not really background staining, but artifacts in the tissue that might look like SARS-CoV-2, and these are labeled by arrows here in red. And you see that there might be precipitates from the antibodies. You see that there might be other precipitates that looks like SARS-CoV-2 but are not. And even if you go to control tissue, and this is also something that we really investigated in great detail. You see that also in people from really uninfected patients that also came to make it sure from like 2018 or 19, before the COVID or the SARS-CoV-2 pandemic started. You see that there are some backgrounds. So we were very cautious to really be able to state what we see is what we think is SARS-CoV-2. And we again went to cell model, and here are virus cells which are highly susceptible for SARS-CoV-2, and you see like lots of infected cells. And if we go to the sushi cells, which is a neuronal human cell line, it's like a tumor cell line, so it's not a super good model for neurons, but we could see that at least some cells could be infected. And I have to say the efficiency is much less than in the virus cells. But with this knowledge, we then went to the brain, and we saw cells that might really carry SARS-CoV-2 in the medulla of persons that died with SARS-CoV-2. However, the abundance of virus-positive cells was super low, and we only rarely saw single infected cells. However, these patients, they are not with acute COVID-19, but they had COVID-19. So this might be also a fact that since we do not see acute disease, that we might also obviously miss some cells. So, but what is known is there are several putative pathways of SARS-CoV-2 into the brain, and one we investigated already in our manuscript. So SARS-CoV-2 might also from the lung, might come via, for example, vagus nerves to the medulla or the spinal cord, to the respiratory center. So this would be one option. The other is obviously that since the virus is also infecting the nasal epithelium, it might also traveling to the olfactory bulb. However, there are manuscripts out there that favor this infection routes, but there are also others that think that this might not the case. So it's still open even after more than 2 years into the pandemic. It's still not super nicely characterized, but also the blood-brain barrier, as I already pointed out, might be a good entry site or for SARS-CoV-2, and it might also lead to vessel dysfunction. So that was also one of our questions. So is there really blood-brain barrier infection, or is it just dysfunction because of overactivation of the immune system? And we first stained with a common marker, which is PKM1 or CD61 in COVID-19 and controls. And when we quantified this, we could see that there's no obvious over difference in the vessels in COVID-19. So we wanted to look more closer to the vessels, and fortunately, I won a nanostring grant for the GeoMx DSP program, so I could work on this digital spatial profiling, which was a super nice opportunity for us. And since you work with slides, as you usually do for your paraffin sections and for your tissue analysis, I was invited to provide 2 slides. So to gain the maximum input, what we did is, so we took regions, and here are some paraffin blocks with brain tissue samples, and did punches, and then we did punches and made this multiple array blocks to get always 6 punches. on one slide and provided 2 slides. So we provided samples of 7 COVID brains and 5 control brains. And we could select 48 vessels. And to sum up this process, so first we nanostring stained the slides with antibody of my choice and hybridized with expression library. Then I could digitally really like live online select the regions where I was interested most. So I checked then selected specific vessels, which I will show you on the next slides. And only in this area, the oligos were cleaved off and then they were sequenced. So we only got the sequence of this, and I specifically used markers for astrocytes, vessels, and immune cells, and DNA as a morphology marker to see the nuclei. And here is in the overview how this looked then. And if you go closer, it was especially nice because in yellow you could see very nicely the vessels, which are also like labeled here. And we specifically chose vessels which did not show so much immune activation, because we really were interested in the epithelial cells and not in the immune cells. And these are some examples and how the regions of interest then looked like, and where these oligos were sucked off then. And as I told you, we selected 48 vessels, and then we compared the expression profiles, and we could really see that certain clusters were specifically upregulated in COVID-19. And so this is also depicted here in this heat map, that really a couple of genes were upregulated, and these clustered especially in groups that were associated with antigen processing and also with interferon gamma signaling, which we thought is super interesting. And so these COVID patients really clustered together, and we then wanted to confirm this further, and we also did qPCR on interferon gamma pathway-related genes, EVTM1 and 2, and they were significantly increased here also in our patients. And I did a staining on EVTM2, and we matched these with patients of the same age to really try to rule out like sampling effects in the age. And it was really super nice that we could also see that EVTM2 is really specific to the vessels, and this we wanted to explore further. Thus, we teamed up with Ole Pless from Fraunhofer Institute, and he just established a very advanced blood-brain barrier model of human iPSC cells, where we really have a tight monolayer where like liquid substances cannot pass. And we added SARS-CoV-2 to the upper wall. So this is really resembling the vessel loom, where versus the lower compartment is resembling the brain parenchyma. And we wonder whether these cells could get infected. And also performing immunofluorescence analysis here for spike and nucleocapsid protein, we could see that these cells could indeed be infected. However, the balance was not super high, so it was around 0.5%. And here is an overview of how it looked. So you again see that not many cells get infected. This is here after 24 hours. And we also did several infections with different virus titers, and we also used different viral isolates just to see whether there's a specific effect on this, but it was not. So we always ended up with about 0.5% infected cells. But what we were wondering whether we could also see replication in our cells, and this is also was investigated by immunofluorescence staining with two other markers, non-structural protein 3, which might be a marker for replication. However, double-stranded RNA is only forming if the virus is actively replicating. And indeed, we could see in the cells that also show nucleocapsid protein a high abundance of this double-stranded RNA. And what we could also see is release of virus to the lower compartment. So we saw an increase here on virus. So we were wondering, are these cells like leaking, or maybe the blood-brain barrier is not tight anymore, so the virus can pass through the pericellular spaces and to reach this basolateral compartment of the cells. And for this, we did electron microscopy on cross-sections of our blood-brain barrier cells. So again, they were infected from the apical side, so it's no wonder that we also see virus attached to the cell here. But we also detected virus in the basolateral compartment. Below, you can still see the membrane where the cells were growing on. And to investigate whether the blood-brain barrier function was still intact, we added fluorescent dye to the upper compartment and then measured if this dye is leaking to the lower compartment. And indeed, after SARS-CoV-2 infection, it was not. So we think that these cells indeed might also release virus to the lower compartment, which would then resemble the brain parenchyma. So in theory, this could mean that these endothelial cells of the blood-brain barrier could pass SARS-CoV-2 into the brain. And we were also interested whether these cells then show the inflammatory profile that we detected with the nanostring DSP analysis in the brains of the COVID patients. And we performed two PCRs also for the interferon gamma related pathway genes, and indeed we could also see significant upregulation in our cells. And this was interesting because, I mean, if you do the DSP in the brain, you also might grab, like, for example, astrocyte and feeds, or also investigate just by chance one or the other inflammatory cell. But this here was a pure iPSC model, so there were no immune cells in this assay. So, and this was interesting that the endothelial cells by themselves can acquire an inflammatory profile by infection and by contact with SARS-CoV-2. Yeah. So we were also interested to check upon the receptors, and we could see that the main receptors, mainly like ACE2, but also NRP1 and the co-receptor TMPRSS2, were indeed also infected in the blood-brain barrier cells. And we were also interested to see whether this could be used for pharmacological interventions. And so we used different methods. So we used an anti-spike antibody, we used anti-ACE2 antibodies, anti-NRP1 antibodies, and nafamostat, which is a TMPRSS2 small inhibitor. And interestingly, as you can see from the immunofluorescence pictures here, an anti-spike antibody So an antibody which is like virtually covering the virus really led to complete blockage of infection, whereas ACE2 and nafamostat had very good results in reducing the amount of infection, and also NRP1 reduced infection. But given the abundance of the different receptors, and I mean, although ACE2 is abundant, it is very low, and still blocking this receptor was highly efficient, as you can see on this quantification here. So this is also really a point that even cells that only express very low amounts of ACE2 might be, for one thing, susceptible for the infection, but might be also saved by pharmacological interventions. And to sum up everything, so we showed that in theory, endothelial cells can be infected, and they could pass the virus to the other side, so the putative brain parenchyma. However, even if this might not be the effect in vivo, still there's an effect on the expression profile of the endothelial layer, namely an upregulation of the interferon gamma pathway, which could be targeted also with effectors that block viral entry. And as Elizabeth already mentioned, our manuscript is the 100th manuscript with nanostring spatial profiling. So it's also a jubileum, so very nice. And to sum up, as I said, so the spatial transcriptomics showed that we have upregulation of the interferon gamma pathway in COVID-19 patients in the neurovascular unit. And this can be also seen in the iPSC-derived brain capillary endothelial cells when they are infected with SARS-CoV-2. SARS-CoV-2 can replicate in these cells and can release virus to the basolateral side, and this can be inhibited by several means, like antibodies, but also small inhibitors. But still, we do not know much about the long-term consequences of SARS-CoV-2. So really, a lot more research is necessary, and a lot more to really understand if SARS-CoV-2 is really entering the brain, how it is entering the brain, and how the abundance or the distribution might be in the brain, or alternatively, how cells are altered in the brain by either direct infection or contact with SARS-CoV-2 or contact with immune cells. So there's a lot to do. And also, since a lot of people also now with the Omicron variant, and I'm talking not about literature here, but also on life experiences with colleagues and friends, that this is really quite common after infection, even in mild infection, that you are more forgetful, that you are very easily tiring, that you feel like easily dizzy after infection. So it's really worth to study these effects in more detail. And with this, I would like to thank my collaborators in the neuropathology, which are mainly Christine Hartmann and Edda Thies. And I would like to thank Ole Pless from Fraunhofer and the collaboration with a super nice blood-brain barrier model, but other also other collaborators in the UKE, like Susanne Krasemann and Fabian Heinrich, other co-workers in Charity. And I would also like to thank NanoString for giving me this nice grant to perform the spatial profiling in the COVID-19 virus, because it really paid, and we have now a much better idea what's going on in the brains. And now we are open for your questions. Thank you very much.
Hello, everyone. Thank you for watching the webinar presentation. So now it's time for the live Q&A, and I know that you have a lot of questions that you're dying to ask Dr. Krasemann. So please go ahead and submit your questions through the Q&A window, and we will commence the Q&A. I actually have a question for you to kind of kick things off. What is the next step in your research in this particular project?
Well, that's actually a good question. So there are a lot of things we still really do not know. So one obvious thing is, I mean, we rather did not directly detect virus in vessels in our patient samples. And I mean, it's a like special cohort, since these are all patients that often had long duration times. So obviously, everything that had happened might not be visible at this time point. So what we really would like to do is also to investigate more brain areas. So until now, we focused on certain areas like our olfactory bulb, which is also the obvious entry route from like the nasal cavity and the olfactory mucosa. We investigated the medulla oblongata, because this is the obvious part if you think that virus could enter the brain via also the virus nerve. So it's also, I mean, we are like focusing on the blood-brain barrier, but we are not restricted to it. So I would really like to check other areas that are not so like obvious, and investigate these, because I mean, we have the material there, and it would be very interesting. Also now that we have, and there are also like papers coming in like every day on COVID. We know some genes that are upregulated, so this would also be maybe a good starting point to check for these genes if they are expressed at specific locations within the brain, and to then start a search in this area. There was just I think some days ago also a very nice paper about neuronal process degeneration in the olfactory bulb in COVID. So also something that even after like more than 2 years, people did not detect yet. So I mean, they used also very nice techniques, and I think there's still a lot to do.
Okay, it looks like we had a question come in. Does SARS-CoV-2 increase protein aggregates in the brain?
That's a good question. However, we only have focused on some brain areas so far, but these do not show like protein aggregates. And I think it's really difficult to tell. That's probably something we would rather expect after like long COVID or so, because also like aggregation of proteins in the brain takes time. And what we know yet is that there's also an upregulation of immune infiltrates. It's not in all patients, but in some. And upregulation of like brain immune cell activity is also going hand in hand with like other things like cytokines and maybe also more phagocytosis, or maybe also more aberrant pruning of neurons or so. And I could imagine that this could maybe alter protein aggregates in the brain, or maybe just speed up what is already so, like in the pre dementia phase, there are also already aggregates in the brain. However, these people do not have dementia yet. And maybe this immune activation might speed up these processes. So I can think of this also to study this in future approaches. It's definitely interesting.
Thanks, and that's a great question. Yep. Would anyone else like to ask a question? Now is your chance. So we had another question come in. Have you thought about doing any single cell sequencing studies or any single cell spatial analysis?
That's also a very good question. But no, we have not. It's also a problem of the sampling process. So, I mean, in Germany, it's also a very strict rule how to handle corpses. And as I said in the beginning of the talk, in the beginning of the pandemic, the city of Hamburg, so the state really ordered autopsy to try in the wish to try to understand more what's happening in these severe COVID-19 cases. And there we had also post-mortem time, but now it's rather also difficult to get autopsies done on COVID patients because it's unfortunately it's so common, and I think this is a big obstacle, especially like in Germany. So post-mortem times increase now. We usually have like 2 to 3 to 5 days of post-mortem. Now, don't think that this is a very good prerequisite to do a single cell, but it would be super interesting. Yes, I agree. But there's a nice story out from the Viscori lab where they did this. It's not single cell; it's single nucleic, and it's also only focusing on some brain regions. But it was published, I think, November or December 2021, and this is really a nice story. I can only recommend reading it.
Great. We had another question come in. After all the testing that you performed for spike and nucleocapsid for antibodies, do you have a couple that you could recommend?
Oh yeah, I have. We currently, I mean, as you may have seen, we currently have a manuscript on this in revision. And what we really realized is that, I mean, that's also obvious from the virus life cycle, that the abundance of nucleocapsid protein in an infected cell is much higher than the abundance of spike protein. And this is not only visible in like freshly infected cells, this is also visible in all the tissues that we studied for the abundance of virus-infected cells and viral proteins. So I can only recommend nucleocapsid antibodies because they give a much better signal-to-noise ratio. And here, I mean, there are several that I could recommend. There's the one from, I think it's xenobiology. It's called MM05. That's a mouse monoclonal. That's very good. And another company, Synaptic System, has two anti-nucleocapsid antibodies in their portfolio, and they really work nicely. And we already tested them on different variants of concerns, including like Delta, and then also including Omicron. And also here, spike protein antibodies often failed because of the variability of the spike, but these nucleocapsid protein, as I said, from synaptic systems, really did a very good job.
Excellent. So another question that came in, does SARS-CoV-2 infection influence the glucose uptake by the brain?
Oh, that's a very good question, but we have not investigated this. I mean, so I can't really tell you, but I think it's a super interesting question, and people should definitely check it, because it might also tell you about vessel disturbances and about what might happen also in long COVID. Maybe there's also some metabolic problems then arising in the brain, and this would be definitely interesting to study.
Thanks. Another question, what happens to drug effluxing transporters in the blood-brain barrier during SARS-CoV-2 infection?
Also an excellent question, but we have not studied yet this. So as you may have seen, to study the blood-brain barrier in vitro, I teamed up with Ole Pletz from Fraunhofer Institute here in Hamburg, and we are currently thinking of different things that we would like to study in this in vitro system before we actually then go again to the brain. And I think this would definitely be something to study more. And also what we see, and this was also, I mean, you might recall the images that I showed, that if you have like the blood-brain barrier model, there are only some cells infected in this, even in this model. And this also, if you take increasing amounts of virus, this is also like saturating. So it's not that you can increase and increase and increase the amount of cells. So it would be also interesting to check whether this specific cell also have a certain profile. So coming back also to the single cell experiments, maybe this would be a more like easy to target approach, and will also tell you something about this molecular or events that then are taking place in these cells. Mm-hmm.
Excellent. Thank you for that question. So we're wrapping up here. If anyone has any additional questions they'd like to ask, feel free to get them in now. If we don't get a chance to address your questions, then we will follow up individually with each of you. So I'll give a couple minutes for last minute questions to get in. All right, I think that concludes the Q&A portion of the webinar. So thank you so much, Susanne. It was a pleasure to have you speak, and thank you to everyone who attended and listened to your talk.
Okay, thank you for listening to the talk, and bye-bye. See you again.
Bye, thank you.
Bye.